Hi, I'm Mitch Drum, the Director of the Research Institute for Children's Health. I'd like to thank you all for joining us this evening for our event. And we thought we'd take you on a virtual tour through the laboratory and show you the types of things that we do and where the funds that are raised through these kinds of events, where they go, so you can better understand where your money is going. I'm in the middle of one of our tissue culture rooms right now, and this is the first step in our process. When we're working on a genetic disease, our ultimate goal is to try to develop therapies and hopefully even cures at some point for these diseases. In our cell culture facility, we can grow human cells in a dish. This cabinet is kept at body temperature. The cells are bathed in a fluid that basically functions the way your blood does. It brings the nutrients to the cells. We pump into their gas so that they get the same oxygen and carbon dioxide that your body cells would. And because these cells are growing outside of the body, they don't have an immune system. So we have to grow them then in these culture hoods here that are kept sterile so that the cells don't get infected and can grow bacteria, fungus, and things like that in them. The job of the cell, it's a factory that makes these different proteins. And each gene in the cell makes a different protein. So you've got about 20,000 different ones. So that's really the number of different diseases that you can imagine could happen. So what we need to know is where the defective protein goes in the cell. We can then put those cells under a microscope like this one. It's a very, very high-powered microscope that allows us to look at very, very fine detail. And I'm pointing out this microscope in particular because this is an instrument that we were able to purchase because of the funds raised by the Research Institute for Children's Health. So events like this have helped support us to be able to do this. Here what we do is we take everything we can get our hands on and try everything and see if anything works. And if it does, then we work backwards to figure out how it works. So what I'm showing you right here is what we call a microtiter plate. So this is basically the same thing as the big plate that we grow cells in. There's about almost 400 on this plate here, so we can grow the cells in there. And then they'll go on to this machine, this robotic machine that will dispense a different drug into each one of those wells. So once we put it in here, the computer will assess every single one of those wells and see if anything's changed. And then you can go back and look and say, hey, wait a minute, B27 did something interesting. Let's go find out what drug that was and what it might be doing.
Part of my research involves examining actual methods to do base editing, and what that means is a fancy way of saying changing the actual DNA structure to fix the primary problem that causes genetic diseases. This is a PCR machine where we can put copies of DNA in there, and it will actually, almost like a photocopier, make many, many more copies of a certain DNA sequence that will enable us to take that, read through the sequence, and look to see if those changes that we meant to make in the DNA were actually present and in what proportion.
So I'd like to finish by thanking all of you for your support over the years and to let you know that the momentum is very, very strong for what we're doing. What you have supported has really been the foundation for lots of other things that have sprung from that. So thank you very much.